All right, I'm really excited to introduce Beibei. Beibei has been postdoc in my lab for a little while, I think. I'm especially excited that she's actually here because most of the time she's been working remotely. She's been in Davis for the past couple of months. Great to actually have her here in person. I met Beibei through a mentoring program as part of the maize genetics cooperation, where I was asked to go visit her poster at a conference. And I was so impressed by talking to her at her poster, I offered her a postdoc right there on the spot. And she has definitely not disappointed. Her background is in epigenetics and genomics. And she's transitioned over the last year or so to working on evolutionary genetics, so learned a bunch of PopGen, and is using sort of population genetics and her past experience in epigenetics and genomics to do some really cool stuff where for the very first time we can actually look at evolutionary genetics of transposable elements on a genome-wide scale. So there's a preprint now up on this after just more than a year in the lab. She already has a preprint, and that's the work that she'll talk about today. Beibei, please.
Thanks for Jeff's introduction. I'm really glad to have this opportunity to share with all of you about my current work, and which is about the evolution of transposable elements in maize. So before we dive into the project, I would like to give you a brief introduction. Just want to make sure we're on the same page. So genetic variation is a basis that evolution works on. But when we talk about genetic variation, most of the time we're talking about a single nucleotide polymorphism, the SNPs. However, transposable elements, which are also called jumping genes, they can transpose from their original genomic location to a new location in the genome. And the transposition of the TE actually can generate other genetic variation, like I show you here, in the deletions and the insertions. Transposable elements was first discovered by Barbara McClintock in maize based on the color difference in the kernels. That's the different color of the kernels actually is due to the transposition of a TE into a gene, which regulates the pigments production in maize. And since then, transposable elements actually has been discovered or identified in almost all eukaryotic genomes. Besides the genetic variation generated by the TE transposition can also generate the different phenotype or drive major evolutionary adaptation. So one classical example is the wrinkled phenotype of the Mendel's pea. This actually is due to the transposition of a TE into a starch branching enzyme. Except the phenotype change, this transposition of TE can also drive evolutionary adaptation. A classical example will be the emergence of the black peppered moth during the Industry Revolution. This is due to the TE insertion into the cortex gene. So besides these effects on the Mendelian phenotypes, the TE and the insertion of TE can also affect the quantitative traits, like I show you here. The P element is another TE. Multiple insertion of P element in different locations in genomes actually can affect the Bristol number in Drosophila. Except this, besides this genetic variation and phenotypic variation induced by TE transposition, the other character of TE is they are really abundant in the plant genome. So here I show you the y-axis is the genome size and the x-axis is different flowering plants, and the red color in the pie chart is how much proportion of genome are TE, and we can see the flowering plants have a lot of TE in their genome, and there is a clear positive relationship between the TE content and genome size. That means TE is a major driver for the genome size variation across the flowering plants. And so we now know TE are abundant in the plant genomes. TE transposition can generate a genetic variation, which can influence phenotypes or affect host fitness. So that makes TE a great system for evolutionary research. And there are some TE evolution research, but most of them actually focus are studied in the plants with really small genomes like the Arabidopsis and the Brachypodium. Their genome size actually much smaller than the average angiosperm genome size. And they have far fewer transposable elements. However, the crops like maize, they have a typical angiosperm genome size much larger than the Arabidopsis, and also you can see they have a lot of T. 85% of the maize genome actually are transposable elements. So this makes maize a really good organism to do the T evolution research. Another reason we want to use maize to do the T evolution because the transposition of T actually causes phenotype change or drop evolutionary adaptation. One example is during the maize domestication, T insertion into the teosinte branch 1 TB1 gene actually caused the increased apical dominance in the domesticated maize compared to its teosinte, which is a major difference between the maize and its ancestor. TE continue shape the maize after the domestication. So the TE insertion in another gene, the MCCT, which is the P fetal period gene, help maize to adapt the temperate environment by altering the flowering time. And another genetic screening, they found that the insertion of the T insertion into a lazy plant 1 LA1 gene actually caused the plants to lose the ability to grow opposite to the gravity. So hopefully now I convince you maize is a really great organism to study the T evolution. But you will ask why people haven't done this and waiting for me to do this. That's because there are some challenges. So this first challenge is it's very hard to accurately identify T insertion using short reads, and that's what most of the research has been using. The reason is T sequences are repetitive, so short read can only cover part of the TE, and the short reads can map multiple locations, so you cannot accurately infer there is a T insertion or not. And here I show you the sensitivity and the precision of T identification using short reads by different programs. And you can see both the sensitivity and the precision are very low. To overcome this challenge, we collect 26 maize genomes publicly available. They are assembled by long reads, and we use one of them as a reference genome and then do the genome alignment between this reference genome and the other 25 maize genomes. And based on the alignment results, we can finally get this T and the SNP polymorphic data I show you here. So in this table, each row will be a SNP or T, and you will have their position and across the population. If there is a SNP or T, the genotype will be 1. If there is no SNP or T, the genotype will be 0. And in total, we got around 20 million SNPs and 130,000 transposable elements. Well, there is another challenge for TE evolution study, is which method we should use to measure the selection on TE. One common method that people use to detect selection on SNP is the SFS, the set frequency spectrum, which describes the distribution of allele frequency across sites in the genome for a particular population. Well, I'll show you a really simplified example to demonstrate how the SFS was constructed and how to use it to detect the selection. So imagine you have a population with 4 samples, 1, 2, 3, 4, and we have some sites have SNPs, the S1 to S6. And you want to use this population to construct a SFS distribution with y-axis is the number of SNPs, the x-axis is the frequency in the population. To do that, basically we need to do for each SNP, we need to count how many samples carry this SNP. So in this example, I show you here, the first 2 SNPs, S1, S2, can see only 1 sample carry each of them. That means we will have 2 SNPs whose frequency is 2. Similarly, we have another 2 SNPs, S3, S4, whose they are carried by another 2 samples. So we have 2 SNPs whose frequency is 2 in the population. Similar, we have only 1 SNP has a frequency of 3, and 1 SNP whose frequency is 4. And in this way, you can construct this SFS distribution. And the reason people use this method to detect selection is because SNPs under different selection conditions, they have different SFS distribution. Their shape change. So for example, compared to the neutral one that I show you here, the SNP under negative selection, they will have more SNPs in the lower frequency groups. That means SFS become left shifted. But under positive selection, there will be more SNPs in the higher frequency group. That means SFS right shifted. So basically, if we use SFS method to detect the selection, people are trying to search signals how the SFS deviates from expected neutral model. This method people also use to detect selection on TE. However, the non-constant TE transposition actually fails this method. That's because one assumption when we use the SFS method is the constant mutation rate of SNP. However, we know T transposition is variable. The one T element may become activated and start amplify spontaneously or under stressful conditions. And then if you compare their SFS distribution between the T and the neutral SNP, you will see the SFS become left shifted, which is really similar to what we got under the negative selection. So this will cause a misinterpretation, whether this signal is caused by T transposition or negative selection. The reason for this is T transposition generate a lot of young TE, and they have really low allele frequency. So one way to solve this problem is we need to match the age between T and the SNP when we compare their SFS distribution. That means when you compare their SFS distribution, you only sample the SNP whose age distribution is similar to the T that are interested. To do that, we need to guess the age of both SNP and T. So we use a program called Relate. Relate basically can construct genome-wide genealogies in the format of trees. Like I show you here, the y-axis will be the time, the x-axis is the genome position. Once you got this tree, you can map both T and the SNP on different branch. And in this way, you can estimate the age. Because we want to match the age between T and SNP, so we divided them based on their age into decile. Just to make sure when you compare their SFS distribution, you got a similar age between T and SNP. After we do the age matching between SNP and T, now you can compare their SFS distribution to search the signal how the SFS distribution deviates or not. To make this more easier, or we want to quantify how the SFS deviates, we calculate the proportion difference between T and the SNP across all the allele frequency bin. And then we only sum those positive proportional difference, and this will be called the SFS. And this is our new statistics to measure the selection on T. And the larger SFS value indicates a stronger selection. So now we got the high quality T polymorphic data, and we also have a method to measure the selection on T. So our goal will be use these new statistics to detect the selection on T across these 26 maize lines. And first, we want to test this method. We want to see whether this method works or not. So we generate some simulated data. So the y-axis is a SFS, the x-axis is a different data set that we simulated. The only difference between them is the proportion of mutations under selected. So P0.5 means half of the mutations that we simulated under selection, and the P1 means all the mutations we simulated are under selection. So our expectation will be the FAS-FS will be increased with more mutations under selected. And consistent with our expectation, we saw the FAS-F indeed increased with more mutations under selection. So now this shows, okay, our method works. We can use this on our real data maybe. And so we first compare the selection on two different SNPs. The high SNPs here are strong deleterious SNP, and the low SNP here are a synonymous SNP. And we can see the strong deleterious SNP has higher FAS-FS value compared to those synonymous SNP. And this is consistent with what we expect. So next, we compare two different class T. These two different class T are divided based on their different transposition mechanism. And we can see the RETE has a little bit higher FAS-FS value compared to the DNTE, but both of them are having a lower value compared to the deleterious SNP, but higher than the synonymous SNP. The two different class T can be further divided into different superfamilies based on what kind of protein they encoded. So the blue bar, okay, the blue bar is the superfamilies from the DNTE, and the red bar is the RNA superfamilies, and the dot is the mean. Sorry, I can see that. Dot is the mean. The arrow bar is the two standard deviation, and we can see there are some variation in the DNA superfamilies with DTA has the lowest FAS-FS, and the DTT has the highest. But for the RNA-T, the superfamilies have very similar FAS-FS. Even though we use T to refer all transposable elements, actually the superfamilies can be further divided into different families, and they are really different in several different characters. So here I show you two most common RNA superfamilies, RLC and RB. Each dot represent a single family, and we can see even within the same superfamily, they have difference in the T length or their distance to gene, or how many of T's are into other T's. And this variation also exists if we look across all the different superfamilies. So this amazing variety actually demonstrates that if we are trying to measure the selection on the T, we need to check on the family level. And that's what we do. And the y-axis is the FAS-FS. The different color on the x-axis is different superfamilies. Each dot is a single family. And we can see the selection is really different for the different families. So now the question will be, what kind of factors contribute to this different selection for different families? If you still remember, when T insert in or nearby gene, they can affect the gene function or expression. So our hypothesis is the T inserted in or nearby gene probably under stronger selection compared to those far away from gene. To test this hypothesis, we split T into different groups based on their distance to the gene. The T is inserted inside the gene. T inserted into the 2 kb flanking region of the gene, 2 kb to 5 kb or over 5 kb. And then we compare their selection. So the y-axis is still FAS-FS. The x-axis is the distance to the gene. And here I show you, for the DNT, you can see the FAS-FS decreased with the increase in the distance to gene. That means a stronger selection for the T in gene or nearby gene compared to those far away from gene. And we got the similar pattern for the RT, but you can also see the RNTE under a stronger selection compared to DNTE at the same genomic location. So now we know, okay, TE insert in and a nearby gene under stronger selection, and the rationale here is because we think TE might affect the nearby gene expression of function. And if this is true, TE should have a very low frequency. And if both these are true, that means the rare TE will enrich with the extreme expression. The extreme expression means with the lowest or highest expression. To test this hypothesis, we use a burden test. Basically, you want to plot the association between the singleton T, that here means the rare T, with the expression rank of the gene. Basically, for each of the single gene, you sort the gene expression across the population, and then you divide the population in different expression rank. So the most left one will be the individuals with the lowest expression, and the most right expression rank will be the individuals with the highest expression. And then you can count the rare T for each gene, and you do this for all the genes that you have. So finally, you will have the total number of rare variants for each expression rank. If there is no association between the rarity and the gene expression, we expect to see almost a horizontal line. But if there are some association or the T can affect either increase or decrease the gene expression, we probably see a smiling plot. To test this, we analyzed the almost over 20,000 genes across the 26 maize line in 10 tissues. But here I'll just show you the results in the answer because they are similar. So here I plot the singleton T insert in gene to see their association with the gene expression. And we can see most of the rare T actually associate with the lowest expression. But we can still see there are some singleton T enriched around those with the highest expression. That means the TE, as long as the TE has learning effects on the nearby gene, no matter it's increase or decrease, they maybe are selected. And we also compare to those TE inserted over 5 kb as a gene, and we can see the association is much weaker. So now we know T inserted in or nearby gene under a stronger selection, and that's much due to their effects on the nearby gene expression. And so the question is how T insertion affects the nearby gene expression. So one way to affect the nearby gene expression is the location that T insert actually is where the transcription factor binding to. So where TF binding to. Chromatin can be divided into closed chromatin and open chromatin based on whether they are transcribable or not. Closed chromatin means they cannot be transcribed, and open chromatin is where TF transcription factor are always binding to and start the transcription. So our hypothesis will be, okay, TE inserted into the open chromatin might under stronger selection because there are potential effects on the TF binding sites. To test this hypothesis, we divide the TE into two groups, those inserted into closed and open chromatin, and also divide them into D and RT. And as I showed you before, the distance between T and the gene can affect their selection. So we need to make sure when we compare the T inserted into closed and open chromatin, they are at the same genomic location. So we first compared those T inserted into closed chromatin for both D and RET. And we can see the 5SFS actually decreased with the increase in the distance. This is the same. This is consistent to what I showed you before when we measure how the distance to the gene affects CT selection. And then we compare the selection to those TE inserted into the open chromatin. And we can see for the DTE, the T inserted in open chromatin actually under stronger selection compared to those inserted into the closed chromatin. And we see a similar pattern for RNA, but much, much weaker. So now we know T inserted into the open chromatin under stronger selection compared to those inserted into the closed chromatin. And the rationale here is they probably affect the transcriptional factor binding sites in some way. And there are some individual examples to show T create TF binding sites. That means the T insertion actually bring in TF binding sites, and the T transcription factor can binding them and regulate the nearby expression. However, there are still some individual examples show that there is TF binding sites before T insertion, and T insertion actually disrupt this TF binding sites, and this can also affect the nearby gene expression. However, we don't know how T insertion affects TF binding sites in the genome-wide, and we also don't know whether this will affect the selection on T themselves. So to test this hypothesis, we got the TF binding sites information across 22 maize lines, and we divide T into basically 3 groups. Non-overlap is a negative control, means there's no relationship between T and the TF binding sites. The create means T insertion might create new TF binding sites and disrupt free. That means the T can disrupt the TF binding sites. And we can see the T insertion both create or disrupt the TF binding sites actually has a higher fast FS value compared to when there's no relationship between T and TF. So for now, we focus on how the interaction between gene and the host gene and the T to affect the selection on T. But if you remember, I showed you T themselves are very diversified in many different characters, like their length, copy number, or their own expression. So we are also wondering whether this intrinsic characters of T actually will affect their own selection. So the first character that we checked is the copy number. There are several different hypotheses about how the copy number of T will affect their selection, but there's no consistent results around this. So we divided the DNA, both DNA and RNA, into the families with the low copy number and high copy number. But for the DNA, I show you here the fast FS for a T with high or low copy numbers, they have very similar fast FS values. But for the RNA T, we can see the T families with high copy numbers may be under stronger selection compared to those has lower copy numbers. Another character we're also interested in is the T length. I don't want to make this really complicated, but I still want to explain. Like the hypothesis here is the longer T at under stronger selection, because the longer T has a high probability to induce the non-homologous recombination, will be really harmful for the genome. So that means the gene, which always has a high recombination rate, maybe has fewer longer T. So to test this hypothesis, we divided both the DNA and the RNA into different lengths. The different colors are different lengths for the DNA and RNA T. And we want to check their length distribution across these different genomic locations. And we can see for the RNA T, there are more longer Ts when they are located far away from the genes. And for DNA, we see a similar pattern, but much weaker compared to what we see here for the RNA. So in summary, we developed a new statistic which can better measure the selection on T based on the age matching between the SNP and the T. And we found that T inserted in or nearby gene may under stronger selection, maybe due to their effects on the nearby gene expression or function by affecting the transcription binding sites. And we also found that some intrinsic characters of T, like T length, can also affect their selection. With that, I would like to thank for Jeff's instruction and support, or thanks to Regina and Nate for troubleshooting the age estimation method, and Manisha help us to get the polymorphic T data, and thanks Natasha to talk with me to make my project and presentation make more sense. And of course, all the lab members in the Raspberry lab, they gave me feedbacks and suggestions about this project and my presentation. And this work is supported by the NSF. And from here, I would like to take any questions and suggestions. We have a preprint, but we still want suggestions and feedbacks.
Very cool talk. I was curious why, so what made me think of this was when you were showing that the statistic was roughly the same for TEs that disrupted and that created transcription factor binding sites.
Yeah.
It just made me think, do you, in your data, is it possible to, not from this statistic obviously, but from like some earlier step to disentangle purifying and positive selection on these TEs? Because I was just thinking like,
okay,
different, like obviously creation or disruption, either one could create positive or negative selection, but it just got me thinking about whether the proportion of those two was different between the two bins. Like if creating or disrupting was more likely to cause purifying selection, and if you've looked at that in other ways, or if you were thinking about looking at that.
okay.
Yeah.
Yeah, I see your point. So you mean like our statistic that as BSFS can both capture the negative and the positive selection, and you want to see whether create and disrupt actually have, maybe there are more positive and negative. We didn't do that for now, but I did try to see whether the T under positive selection on the whole gene, on the genome-wide, we didn't see significant the T on the positive selection. So yeah, and here we think maybe like this actually is really similar that we got from the gene expression, because we think maybe T under, because we always think T is deleterious, right?
Yeah.
They should be under the negative selection. But like we show you here, like the T, the rare T actually can associate with both the lost and the past expression. So we think maybe the T actually under stable lapping selection. Maybe, yeah. That's just yeah, we are still thinking about that, but yeah.
Thanks. I'm very naive about T's, so that's a preface to my question. For the family level effects, which I thought were very interesting, are those explained by some of the other effects you see? Like, are certain families of T's longer? Or right, like I guess you presented them as kind of different pieces, but I assume you've looked at whether they're connected.
So you mean that, okay, so you mean that we have a lot of factors may affect the selection on T, and you see here a lot of different selection on different family. You want to know which factors contribute most, right? Is that
or whether the differences in families.
I also want to know, and also that's maybe knowable that we can just plot their length distribution for the single families or something. My guess is that each single families maybe have the the factors that contribute to the different selection for each single families may be different. We can check that. We can check for each single families to see whether their distance to the gene is different. That's maybe the factor that make they are under stronger selection, or we can check their T length. But I think here we present the distance to the gene may be the most important factor. But when you plot the distance to the gene for each single families, you won't say, oh, this has a lowest BSFS. Maybe they are most of them far away. That's what we want to get. But I don't think you will see, oh, this is any factor. I think many factors.
Yeah, sure.
Yeah.
Thank you.
Sure.
Awesome talk, beautiful, clear visuals. Thank you for describing that in the way that was easy to understand. What do you make of the differences between RNA and DNA TEs? Because they're sort of like, at first it felt, the two first things you said were sort of that the RNA TE families varied less, right? And then that there was no difference between open and closed chromatin for RNA TEs. But then it sort of flipped and DNA was not changing based on copy number variant and TE size, and RNA was, and I just,
Okay.
like, is one consistently larger than the other? Is it something about how there's RNA involved in the copying? I don't know. Oh, sorry.
Okay.
That's fine.
Yes, the DNT and the RT actually they are very different. That's the principle that we use to divide T into DNA RNA actually based on their transposition mechanism. For the DNT, actually you just cut it from its original base and then insert a new one. For the RNT, actually you need to first transcribe into the RNA and then reverse transcribe to DNA and then insert it. But that's not any difference. The DNT are usually very short compared to the RNT. And also, like, I think maybe like that's why we see the results here for the T lens. You will see a very for the RNT here. You can see that.
Oh yeah, the size there.
Yeah. For the RNT, you can see.
Oh yeah.
Yeah. There are more longer T's like over 5 kb. I think there are different hypotheses actually under this different results for the DNA and RNA results difference here. Yeah.
I was curious, this might be a really simple question, but I was curious for you, the SFS statistic you made. I was curious, should it be interpreted as like, is there like some threshold that over which you say that this is like strong selection, or should it be interpreted as a relative measure of like, you look at it between DNA and RNA or between in gene and just outside a gene type thing?
Yeah, I think probably we can compare to this high SNPs. There are that strongly deleterious SNPs, so they are supposed to under very high selection. So basically, we can compare to this and to show how strong the selection are on the T. And for the T families, you can see some of them actually comparable to the deleterious SNPs.
Yeah, so it's better to think of it as like a relative metric, not as like a.
Yeah. It's a relative one, not the absolute value. Yeah.
Right. Thank you.
I guess I was thinking about this deficit statistic, and I mean it's like a shortfall of transposable elements compared to the neutral site frequency spectrum. And so it's like transposable elements that are missing because they've been selected out. And I wondered if thinking about it in that form, sorry, this is not a precisely thought through question or even statement, but like, can you get back to things like the insertion rate through time or the insertion rate of different classes of T along the genome using that approach? And have you thought about doing?
You mean whether there are some T amplification during the time?
Sorry, I missed the.
Did you mean there are some T transpositions along the genome or something? Sorry, I didn't get your question.
Yeah, it was not well posed, but I guess I'm just wondering, like, you're sort of showing it as an empirical statistic, but I wonder if there's some way to interpret it in a deeper way and think about it as a deficit and then calculate what is missing on the basis of that.
Okay. I don't think I have thought about that, but yeah. I haven't thought about that.
Other questions for Beibei?
Just one more question. So this is super cool, but I was thinking about like what other creatures or organisms we have 26 long genomes for. And so, as someone who works on a different creature with very weird, large transposable element filled genomes, do you have any sense of like whether you think, or can you speculate whether these patterns are true across different organisms, or is corn just special? I mean, it's obviously special.
So you mean that if you work on another organism, but you don't have a long-range genome sampling, right? You want,
Well, I guess if you went and did exactly this in another organism, do you think these kinds of patterns are similar across organisms, or do you think it's possible that TEs in different organisms behave really differently? Like that you wouldn't see more selection on longer TEs or
Okay.
something?
I think, as far as I know, at least in some of the plants that they have started the T evolution and use the short reads in the smaller genomes. And they also find that the distance may be a factor that contributes to the different selection. And also, that's the open chromatin, closed chromatin also is the factor.
Thank you.
Okay, let's thank Beibei again.